Greetings, everyone. I'm Liz Fernandez with BS Training, and I would also like to welcome you to the webinar today. Our speaker today is Dr. Vienna Brown. Dr. Brown is a biologist working for the National Feral Swine Program and oversees all activities related to feral swine disease issues. She completed a bachelor's degree in animal science from Colorado State University, a Master of Public Health from the University of Colorado, Denver, and a PhD in microbiology from Colorado State University. And with that, I'm going to pass over the webinar to Dr. Brown.
Thanks so much, Liz. Good morning, everyone, and I'd like to start by thanking you for your time and attention on this webinar, and a special thank you to Liz Fernandez for the opportunity to present this morning. I do have a pretty hefty slide deck this morning, so I'm going to move fairly quickly and get started right away with ongoing disease efforts in feral swine. A bit of background on the seminar overview. We'll start first by talking about feral swine and why they are a tremendous issue, and move into the overall framework of the APHIS National Feral Swine Damage Management Program. Then I'll move specifically into disease-related issues, starting with national disease surveillance, targeted collaborations, a subset of our continuing education opportunities that we provide to our field biologists, and then move into the interface with veterinary services. So, feral swine, swine were first brought to the U.S. in about the 1500s by explorers, and then there have been repeated introductions ever since. In the past 20 to 30 years, feral swine populations have expanded dramatically, and the distribution has also expanded. This is in large part due to anthropogenic movement, as well as escapes from fenced farming and hunting operations and breeding on the landscape. There are believed to be well over 6 million feral swine that are now found across more than 35 states in the U.S. The current feral swine distribution, this is just a present absence map and doesn't demonstrate densities. However, you can see that the vast majority of feral swine populations are found in California and then Texas, Oklahoma, and across the whole Southwest region up into the Carolinas. Of course, there are a smattering of other populations across many of the other states, and those are either due to, again, anthropogenic movement or escapees or releases from hunting or farming operations. Feral swine are hugely problematic. They damage almost everything that they get into. There's almost no agricultural product that does not damage either by consuming it directly or damaging the landscape. They also are hugely problematic for natural resources, including threatened and endangered species, riparian areas, and a number of other issues. They also cause extensive damage to property, historical sites, architectural sites, golf courses, and other properties. And then, of course, they are a risk to humans and other animals, both from a behavioral standpoint as well as a disease transmission standpoint. The APHIS National Feral Swine Damage Management Program was developed in March of 2014 when APHIS received an appropriation for $20 million to implement a collaborative national feral swine damage management program in states where feral swine populations were recognized. The overarching goal of the National Feral Swine Program is to minimize damage caused by feral swine and to protect agriculture, natural resources, property, and human health and safety. The National Feral Swine Program has two major operating approaches. In states where feral swine densities are low or fragmented, we really aim to eradicate swine from those states. And in states where feral swine populations are high and are long established, we work to manage damage. From an operational approach, each state is labeled with a level 0 through level 5, which is used as a proxy for feral swine density. And then that is used to determine baseline funding levels for operational control of feral swine. The darker the color, the higher the perceived feral swine densities are, and the corresponding higher level of baseline funding that is provided. We also do operational control in 3 territories, including Guam, Puerto Rico, and the U.S. Virgin Islands. And I'd like to move into the disease surveillance component, which is the heavy lift for today's presentation. We have been doing national feral swine surveillance from a targeted standpoint for the last 5 years. The first version, there were identified 3 diseases of national concern: classical swine fever, CSF, swine brucellosis, SB, and pseudorabies, which is PRV. We have had a congressional mandate to collect a minimum of 2,800 samples annually from feral swine across the U.S. And this was under a targeted surveillance approach with samples dispersed across both space and time, with counties being prioritized based on existing feral swine populations, domestic hog production, and the presence of landfills. It is important to note that the data roll-up on this gives us an apparent seroprevalence, just looking at the number of samples positive over the total number of samples tested. On the graph on the right, if you notice, between 2017 and 2020 fiscal years, there was a fairly uniform or kind of pockets of seropositivity that was detected. For classical swine fever, quite predictably, we have not had any positive samples. This is a foreign animal disease, and so that was to be expected. For pseudorabies, our seropositivity in feral swine bounces between about 17.5% and 25%. And then for swine brucellosis, seropositivity levels bounce between about 5.5% and 10%. If we look at that data at a nationwide seroprevalence, so that graph was that table was an aggregate roll-up across all of the U.S. where feral swine are being removed and sampled. If we look at it broken down by state, this is a heat map looking at apparent seroprevalence from the aggregate of fiscal year 2020 for pseudorabies. The darker the color, the higher the level of seroprevalence. And so you can see there's a handful of states that have greater than 25% seropositivity in feral swine that were removed and sampled. However, if we break that data down even further and down to the county level, this is the same data. This is again an aggregate of fiscal year 2020 for Texas. Texas at the state level has a seropositivity exceeding 25%. However, if you look at the county level, there are a number of counties that have significantly lower apparent seroprevalence for pseudorabies as compared to the state aggregate. And so it's important to note as we increase granularity on the data, we do see a number of counties that are side by side that have very different levels of apparent seroprevalence. And so it's important to note as we roll up the data and the more aggregated it is, of course, we lose a lot of that granularity, and we don't see necessarily the same levels of infection across the state or even the county level. For swine brucellosis, we see a similar seroprevalence, not as many states with such high levels of seroprevalence. However, we do see, again, if we look at Texas at the state level, we see between 6 and 10% seroprevalence. However, if we break it down at the state level, the state down to the county level, you can see, again, we have very few individual counties that come back with between 6 and 10% seropositivity. We have many states that come back with no positive samples that were tested, and then a number of counties that also come back with greater than 25%. So again, as we aggregate this data, we lose the fine details, and we really do see pockets of very high and very low seroprevalence across the counties in the states. In fiscal year 2019, we rolled out morbidity and mortality surveillance for African swine fever. The morbidity and mortality surveillance is predicated on our field biologists looking for sick and inexplicably dead feral swine on the landscape. We just had our 10th case reported earlier this week. Florida has had 3 incidences of foreign animal disease investigations in California and Indiana. And so these samples are triaged through Veterinary Services. We work in close partnership at the regional level with the Veterinary Services area veterinarian in charge, and they have ultimate authority on whether these samples move forward as a foreign animal disease investigation or not. So these samples are triaged through the foreign animal disease investigation network that Veterinary Services has in place. For the 10 cases that have tripped the trigger, if you will, on a morbidity and mortality surveillance, 5 have gone forward as full foreign animal disease workup, 3 from Florida, 1 from California, and 1 from Indiana, and all have come back negative for both ASF and CSF. I want to walk through one specific case study for our African swine fever morbidity and mortality surveillance. This was in Mississippi. It was a lone boar that was found in April of 2020 to be in extremely poor body condition with a hoof lesion on the right front limb. The field biologist that found this animal contacted the state director and both headquarters staff from both the National Feral Swine Programs and the National Wildlife Disease Program. The Veterinary Services area veterinarian in charge was also contacted and photos were sent. Upon further investigation, the animal was found to have an embedded constricted band of plastic around the hoof, and samples were not sent forward as a full foreign animal disease workup as per guidance from the Veterinary Services Agency. However, the carcass was incinerated at Mississippi State University Diagnostic Lab as just an extra layer of precaution. And so this example just really demonstrates the interwovenness of wildlife services with other federal agencies and state agencies, and then on-the-ground collaborators such as Mississippi State University in this example. The National Cross-Line Damage Management Program did roll out a version 2.0, if you will, of national surveillance at the start of fiscal year 21. This is predicated as a foreign animal disease surveillance system. It's particularly focused on three major foreign animal diseases at this time: tropical swine fever, African swine fever, and foot and mouth disease. The surveillance approach is to continue doing active surveillance for tropical swine fever, in that we're taking blood, sending it down for serum, and sending that off the saddle to look for antibodies. We will continue doing morbidity mortality surveillance for African swine fever, and we're doing what we call active observational surveillance for foot and mouth disease, where we ask our biologists to look in the mouth, along the tongue, the gums, the nose, the snout, and along the host for any evidence of vesicular lesion. For both ASF and FMD surveillance, any concern around either of those diseases will be again triaged through the Veterinary Services ABIC, and those samples will fall through the foreign animal disease investigation workup from Veterinary Services processes. The primary risk factors that have gone into this new model are related to introduction risks and host risks. The introduction risks, the heavy lift on that weight of that component of the model is using airline passenger data. This has been in close partnership with Dr. Ryan Miller and Dr. Garrica Cook out of SIA, and a data set from Customs and Border Protection looking at products that are illegally imported and confiscated. So this data set is able to determine where passengers were starting and where they're ending, and if a product is confiscated, what that was. And so we can then move backward and set up a list of countries that are endemic for the three diseases of the three foreign animal diseases of concern, and then determine where the risk would have been if the product had not been confiscated. So that's really an impressive data set, and it's been really helpful. We also are using the presence of landfills and seaports of entry to inform introduction risks as part of the weighting for each county. Host risks are twofold. One is around domestic livestock populations. So for ASF and CSF, we are using presence of domestic swine production exclusively, and then for FMD, we are weighing all host stock production. And then, of course, presence of feral swine populations are an important component of host risk. And so the compilation of each introduction risks and host risks, and then each of the subset of those data are then worked up to provide an overall county prioritization. It is important to note that it is still within an opportunistic framework. So feral swine are being removed operationally as a part of damage management, and so we are however guiding our field biologists on where to do the majority of their sampling, which of course would be in high-risk counties. Something that's very exciting about this new version is that it can be easily updated to address emerging threats. So when countries either break with new diseases or eradicate, if they have an endemic disease and they're able to eradicate it, that can be worked in to modify the risk of introduction. We can also change readily with changing APHIS priorities or new emerging diseases. Importantly, this new surveillance metric does integrate monitoring of production diseases. So we are continuing to use the serology for both pseudorabies and swine brucellosis. However, importantly, these neither PRB nor SB are being used to weight the counties. It is just this surveillance effort is being piggybacked on top of the foreign animal disease efforts. We also are very excited that we are down to the 1% detection rate, which of course is in line with OIE standards of surveillance, and this has nearly doubled our sample size to nearly 6,000 animals annually. And this is an important time to stop briefly and thank our field biologists and state directors and our trappers and technicians that are out collecting these samples. It is not a trivial effort to collect these samples and get them shipped off and triaged to different laboratories. It's a huge lift, and we've had tremendous support and buy-in from our biologists, and so thankful for that. With that, I'd like to move into targeted collaborations. And so these are projects that are ongoing that are brought to us by other stakeholder groups. So the National Cross-Line Program is really intentional about working closely with state directors, working with state agencies, working with other APHIS agencies and our different stakeholder groups often bring projects to us. Hey, what about this in feral swine? What about anthrax? Could feral swine be involved in that? What about parasitic infections on the landscape? What about CWD? And so when those projects come up, we do our best to work with in-house researchers, National Wildlife Research Center researchers. However, sometimes the expertise is not necessarily in-house, and so we will work specifically with university groups to get those important questions answered. So I will walk through a number of these targeted collaborations. Keep in mind this is not a comprehensive list, but should provide kind of a great perspective of the type of work the National Feral Swine Program is involved in above and beyond national surveillance efforts. The first project I'd like to talk about is chronic wasting disease. This project came to the forefront following a publication that came out in 2017 demonstrating that swine are capable of becoming infected with chronic wasting disease prions following experimental exposure. We know that we have a number of areas across the country where there are very high levels of feral swine that are cohabitating with cervids that are often infected with chronic wasting disease. Newton County, Arkansas rose to the top of the list at the start of this project, where the prevalence of chronic wasting disease in cervids exceeds 25%. And so we worked in close partnership with Clint Turnage and a number of others in state agencies and on the ground to collect adult feral swine in this area. We do know from a behavioral standpoint that feral swine will consume cervids that have died from chronic wasting disease on the landscape. So there was certainly a behavioral component and a biological component to think that interspecies transmission was at least a possibility. So as far as the original onset of the project, 102 adult feral swine were collected from Newton County, Arkansas, and we collected a number of feral swine from Dewey and Caddo counties, Oklahoma, to serve as negative controls, because this is believed to be a CWD negative region. And again, this project was a tremendous work from our field biologist. Clint Turnage really spearheaded the efforts in Arkansas, and then Pat Whitley worked on the feral swine in Oklahoma. And we just recently started a third branch of this project in Texas, based on a newly emerging chronic wasting disease problem in cervids. A huge plethora of tissues were collected from these animals: brain, a smattering of lymph nodes, whole blood, GI tract, tonsil, feces, and masseter muscle. And this has been a hugely collaborative effort. We worked with Iowa State University for the RT-QuIC, and then University of Texas Health for the PMCA assay. Colorado State University conducted the immunohistochemistry, and then any animal that had a multitude of samples that came back as either suspect or probable positive were then put into a mouse bioassay, which of course is the gold standard. Transgenic mice, both cervidized and porcinized mice, were inoculated with suspect samples in the amplification assays. And of course, as you can imagine, this is a hugely complex study. Interspecies transmission of any pathogen is a huge concern, of course, and also fairly difficult to kind of track in a wild population. And so, very little has been done on feral swine in general related to chronic wasting disease issues. So we have had to be really intentional about results interpretation. So preliminary results in vitro, so these are using RT-QuIC, PMCA using the deer substrate, and then PMCA using a pig substrate, as well as IHC. You can see, so for pigs number 49 and 59 that came out of Arkansas, as you follow that across horizontally, you can see for animal 49, it came back positive by RT-QuIC from a brain sample, probable positive brain sample, and from the submandibular lymph node using PMCA and the deer substrate, and then the retropharyngeal lymph node using porcine substrate. And same for animal number 59, you can see a number of weak positives or positives, suspect positives as you move across the scope of diagnostic assays. So these two animals in specific, as well as several others, were put into a transgenic mouse model to evaluate the prion status in those samples. In vivo, we've seen a number of really interesting results, really preliminary. We did infect 10 transgenic mice with brains from 2 different animals, submandibular lymph nodes from one animal, and the retropharyngeal lymph nodes from a different animal. 4 of the mice that were infected with brain samples from animal ID 49 were found dead right around 100 days post-challenge, which of course is very early on for chronic wasting disease, prion infections in general. Two mice that were infected with retropharyngeal lymph node samples from the other feral swine were found dead right around 100 days as well. Interestingly, these were all female mice, and they were porcinoized transgenic. They did develop neurological signs and go on to die after developing atypical nesting activity, poor grooming, and hyperactivity. Currently, there is no evidence of prion disease in these mice. We have not been able to find it using either PMC or Western blot assays, but we are certainly intrigued that these animals are developing neurological lesions or atypical neurological behavior following potential infection with chronic wasting disease in feral swine. Of course, the implications of chronic wasting disease in feral swine are tremendous, which is why the National Feral Swine Damage Management Program is invested in this stakeholder question. Interspecies transmission of pathogens is, in general, hugely problematic and very distressing. There's also a question that maybe feral swine are not becoming infected with CWD, but they are, it is passing through them, and there's a potential for prion dissemination across the landscape. And of course, anthropogenic movement of feral swine is hugely problematic, and there's certainly a possibility that if feral swine are implicated in chronic wasting disease, either becoming infected or serving to transmit the prion, that these intra and interstate movements are even more problematic than they currently are. So this project is it's slow growing in the sense that to call mice negative, we have to go out 500 days post-challenge, which is about a year and a half time. And so this has been quite a complex project. It's been, there's a number of collaborators, and we're just kind of waiting to see how this all shakes out. But we are quite invested in the world of prions. I'd like to move into another targeted collaboration. This is a Puerto Rico eradication project. Following the Hurricane Maria in 2016, the feral swine have become hugely problematic. These aren't even really feral swine necessarily in Puerto Rico, but just urban swine. You can see from the pictures that they are in neighborhoods, on playgrounds, outside children's schools, and in the streets in large numbers, and quite accustomed to humans. And so this is quite problematic. Puerto Rico, as you may well know, is an island off the coast of Florida, but its close neighbors, Cuba, Haiti, and Dominican Republic, all have had issues with both ASF and CSF in the last several decades. There is a huge amount of movement of personnel and products between these islands, and the introduction of a foreign animal disease, of course, to Puerto Rico would have tremendous implications for our trade relationships nationwide. So we're using this opportunity to remove large numbers of feral swine in these urban environments, and we're really using that as an opportunity to cut our teeth on doing antigen-based diagnostics in apparently healthy animals. All of our ASF work currently, from a national surveillance perspective, is related to sick and dead animals. However, in Puerto Rico, we are going to be doing PCR on apparently healthy animals. We are also doing PCR for both CSF and influenza in these swine that are removed out of Puerto Rico. This is a great opportunity to see not just historical exposure, which is the majority of what we see for national surveillance efforts, but to also have an understanding of incidence of disease in these animals using antigen-based diagnostics. We will, of course, continue doing national surveillance efforts in these animals as well, doing serological diagnostics for CSF, SB, and PRV. Another targeted collaboration we have ongoing is related to brucella suus inoculation. Swine in general, serological diagnostics chronically underestimate true disease prevalence for infection with brucella suis, which is the causative agent of swine brucellosis. And so because we do a tremendous amount of work at the national scale looking for swine brucellosis using serological diagnostics, having a robust understanding of how serology fits into how serological diagnostics and our national surveillance at large fit into true disease status has been an important question. So we have partnered with Colorado State University to do an experimental inoculation in their large animal BSL-3 facility. We went on to inspect 24 feral swine proxies, what we're calling them. We used off-the-island swine that came out of North Carolina from a breeder that has been breeding these animals for several decades and no known exposure to or issues around brucellosis infections. They also came back all seronegative to begin with. We infected these animals with one of two strains, either the reference isolate, which is strain 1330, or a field isolate. We wanted them to euthanize these animals serially at 1, 2, 4, 8, and 12 and 16 weeks post-infection, and collected a number of tissues at necropsy. All surviving pigs were bled every 4 weeks, starting at 4 weeks post-infection. We have repeat bleedings from these animals over time. From a culture standpoint, tissues were collected and cultured on brucella-selected media, looked at blood, vaginal swab, endometrium, lymph nodes, and then spleen, liver, and lung. In general, the lymph nodes and spleen have the highest concentration of bacteria, and specifically cranial lymph nodes were one of the best tissues choice for culture. Bacteremia was present at the time of necropsy in nearly 40% of pigs, and the number of positive tissues per pig was highest between 2 and 8 weeks post-infection, but even 4 months out, all animals had at least one positive tissue. From a serological standpoint, I'd like to walk you through this graph. It's a little bit hefty, so bear with me on this. The x-axis is the week post-infection that the animal was euthanized, and the y-axis is the number of pigs that were tested at each time point. The green are animals that came back positive, purple is animals that came back negative, and then we used 4 different diagnostics for each animal at each time point. The card test it does not have any polka dots. It's just a solid graph. BAPA has the largest and furthest spaced polka dots. Complement fixation is the next largest and next furthest spaced, and then FTA is the smallest polka dots that are the closest together. So if, for example, you look at week 4, using the card test at week 4, 1 animal came back negative and 15 animals came back positive. If, however, we look at the FTA diagnostic at that same time point, so these are same animals, same infection, same everything, same samples, 50% of animals came back positive, 50% came back negative. And so this was really interesting to see, also a little bit concerning from a national surveillance standpoint, but you know, using these animals with known infection status and known challenge, we were able to kind of begin to elucidate those results. One animal that I want to draw your attention to in specific was if you go up to 16 weeks post-infection, you can see that there's one animal that did not seroconvert. That animal that has the purple, one animal has purple bars on all 4 diagnostics. That animal did not seroconvert over the entire study period, and so you're probably thinking what we were, which is perhaps this animal was never infected. But if you go back to the culture results, that animal, we were able to culture live bacteria from a mammary lymph node. So that animal was infected and just never demonstrated a detectable antibody response across the study period. So what we have seen from wild infected animal or feral swine on the landscape matches what we see in a laboratory setting. So that was a really, really neat finding. Another targeted collaboration we have ongoing is related to leptospira in both feral swine and water. This is in partnership with Auburn University. Leptospirosis is a zoonotic bacterial disease of global importance, and Auburn University has been really involved in research related to lepto for many years. They've developed a highly sensitive assay for detecting pathogenic leptospires, which is 100-fold more sensitive than the current accepted assay. They went on to evaluate water, springbed sediment, tissue, and blood samples from feral swine. Another ongoing collaboration we have is with Squidix, which out of the University of Georgia, looking at 3 important parasitic pathogens of concern: Toxoplasma gondii, Trichinella species, and Trypanosoma cruzi. These are Toxoplasma and Trichinella species have both been looked at in previous data sets and related to feral swine prevalence of these diseases. And this current project would expand the data sets from samples collected between 2007 and 2014. We aim to test samples from a later time frame to evaluate prevalence over time in previously sampled regions to see if it stays the same or if there's any fluctuations in that feral prevalence. We also aim to test additional samples from previously under or unsampled regions, and then use statistical analyses to determine what, if any, epidemiological factors are associated with infection in feral swine. Trypanosoma cruzi is also an important zoonotic parasite, and this project is a pilot project really just aiming to evaluate if feral swine become infected and if they may play any sort of role on the landscape. We've also partnered with Colorado State University to look at do some flavivirus surveillance in feral swine. In 2015, there was a widespread Zika virus outbreak in many parts of the world, but including Puerto Rico. Typically, Zika virus is associated with an asymptomatic infection or very mild clinical disease. However, that 2015 outbreak was associated with very severe birth defects, including microencephaly and others in the fetus. In 2019, Wildlife Services operations were doing a large-scale removal of feral swine from San Juan, Puerto Rico. Serum samples were collected from 250 feral swine, and in partnership with CSU, a LAMP assay was used for detecting both Asian and African lineage Zika viruses. These assays also have an additional ability to look further for flaviviruses, including chikungunya, West Nile virus, and dengue. To date, 200 samples have been tested and none have come back with antibodies for Zika virus. We are also working in close partnership with NWRC researchers, Dr. Alan Franklin and his colleagues, to look for evidence of antimicrobial resistance in feral swine in the San Francisco Bay Area. This project, the objective is to estimate the potential exposure of feral swine at frequent landfills to bacteria resistant to colistin and other high-priority antibiotics. Surprisingly, there's a number of feral swine that live in the San Francisco Bay Area and frequently feed at these landfills. And so there's tremendous concern about exposure to foreign animal diseases as well as antimicrobial resistance. From a methodological standpoint, we are sampling fecal matter from 100 feral swine that are removed by Wildlife Services operations from these landfills, and the samples will be analyzed for evidence of resistance to colistin and other high-priority antibiotics, including carbapenems, third-generation cephalosporins, and fluoroquinolones. To date, 17 samples have been collected, and all of the laboratory detection methodology has been validated. We are also doing a project with Colorado State University related to bacillus anthracis infection. In the summer of 2019, Texas had an extremely bad anthrax outbreak that fell outside of the normal endemic county region and was particularly severe. Cattle, sheep, goats, and both native and exotic deer species were infected, and anecdotally, feral swine moved into that area about 3 to 4 years ago. This was a project that was brought to us by the Texas Wildlife Services State Director, wondering about the potential role for feral swine contributing to bacillus anthracis presence and transmission on the landscape. So again, we have partnered with Colorado State University. The access to their BSL-3 facility is invaluable, as well as their expertise and collaborative nature. The goal of this project is to obtain baseline information on susceptibility of young feral swine to inoculation with anthrax spores and to characterize both their pathological and their immune response over time. We will be collecting 20 young feral swine from anthrax-free region of Texas and moving them into the large animal BSL-3 facility for infection. This will be, again, a serial euthanasia, and we'll be following these animals out over time. Another targeted collaboration ongoing with Colorado State University is related to coccidioides exposure in feral swine. Coccidioides is a pathogenic fungus that causes valley fever, and these spores can be inhaled by susceptible mammals, including humans, with the disease often presenting as a persistent pneumonia. This fungus is thought to occur primarily in an arid desertic region, and although the ecology is thought to be tied primarily to desert rodents and burrowing animals, susceptibility to other species remains a question. So in partnership with Colorado State University, we are aiming to develop a serological diagnostic that can be utilized to determine feral swine exposure to coccidioides species. We can also intend to do an experimental infection to document the immune response of feral swine to various levels of coccidioides species spores over time and by different routes of exposure. This will allow us to document pathogenesis, fungal load, and fungal shedding in infected feral swine. Of course, the overall implications of this type of work is to determine if feral swine do demonstrate a measurable immune response to coccidioides species, they may be useful as a biosentinel for this pathogen on the landscape. And if feral swine do become infected and shed fungal spores, they may be contributing to pathogen transmission on the landscape. And of course, the issue of anthropogenic movement is problematic for a number of reasons, but of course, pathogen dissemination. We are also partnering with Auburn University and ARS to look at bovine viral diarrhea virus in feral swine. BVDV is a hugely important viral pathogen in U.S. cattle, causing tremendous economic impact due to morbidity, mortality, and production losses. Domestic swine and Eurasian wild boar have both been demonstrated to develop evidence of infection, and so we were working with him to screen feral swine serum samples for presence of antibiotics. Other work that we have ongoing with Wildlife Services researchers out of the National Wildlife Research Center is related to GWAS, and this is a genome-wide association study. If you remember briefly from your genetics classes probably decades ago, the observable characteristic or the phenotype is a product of environmental factors and the genotype, which is a genetic makeup. The single nucleotide polymorphisms are what we're particularly interested in relation to these projects. So in the cartoon depiction, we are not interested in the T and the A and the G and the G and the C, where the human genome lines up. We're really interested in the nucleotides in the box, the A and the G that do not match up. And so SNP is a DNA sequence variation occurring when a single nucleotide differs between members of a species, and you can use this to determine if there's either in any sort of relationship with, in this case, the disease phenotype that we're seeing in relation to the phenotype. And so we really began to cut our teeth on this. We have one project that is nearly completed, one project that's in progress, and one project that is just starting, looking at the genetic analyses in relation to the study of feral swine disease. And Dr. Tony Fiasio, Dr. Tim Smither, and Courtney Pierce have been really invaluable in conducting these projects. So the project that we've recently completed is around the culture and serological results associated with swine brucellosis infection. So prior to my tenure, Wildlife Services collected 376 both paired blood and tissue samples from feral swine that were in an abattoir in Texas. Of those animals, the 376 that were collected, 49 of those animals were culture positive for Brucella suis. 16 of those animals were seropositive, and 33 were seronegative. And so we used the animals that were culture positive, seropositive as the cases, and the animals that were culture positive, seronegative as the controls. And then we can use this genome-wide association study to see if there's any sort of relationship there. And so if you look at these Manhattan plots, you see the significant loci fall above the line of significance. And so using the additive, dominant, and recessive models, we came up with 9 loci that were significant. As an example of how these results are interpreted, one example was the Elmo gene came back as a significant sample. And this SNP was associated with a decrease in seroconversion. And so the hypothesis around the potential relationship between the genotype and the phenotype is that the ELMO1, a modification of the ELMO1 gene may decrease humoral response because it reduces internalization of Brucella suis by phagocytic cells. This then reduces the pro-inflammatory immune response associated with bacterial uptake, leading to an overall decrease in seroconversion. And this work was done also in partnership with Steve Siebelson and Paula Fogiato out of ARS. And so in terms of results, interpretation, and implications, all 9 of the candidate genes that came forward as genes of interest for the GWAS study were related to immune function. It is important to note that these results are difficult to interpret and do not demonstrate a causal relationship. This is really used as a hypothesis-generating tool. However, from an operationalization standpoint, it does allow us to identify populations that may have a poor serologic response following Brucella suis infection, and it can allow us to better target our surveillance efforts, making them more effective. We could steer clear of doing any sort of serological diagnostics in animals that have a genotype associated with decreased seroconversion following infection. This would minimize our risk of false negatives and also could be used to inform risk assessment. We also have just started on a genome-wide association study in partnership with the NWRC Genetics Project, as well as researchers out of Makerere University in Uganda, to evaluate the genetic underpinnings of African swine fever virus tolerance and susceptibility. A number of genetic samples have been collected from swine in Uganda with known African swine fever virus exposure, including domestic swine, bush pigs, and warthogs. We are using a high-density genotyping array with 660,000 loci. And these findings, again, may be operationalized in the event of an African swine fever outbreak in the U.S., where feral swine genotypes could be utilized to guide management practices. We also have an ongoing targeted collaboration with the Economics Project out of the National Wildlife Research Center. This is in close partnership with Dr. Stephanie Schwitz and Dr. Ryan Miller and Dr. Sophie McKee, where we're really working to understand the potential economic impact of U.S. agriculture from feral swine-mediated foot-and-mouth disease, African swine fever, or classical swine fever. This project is broken into 3 primary phases. The first phase is complete and is a risk assessment with the 2 primary questions being around worst-case scenario and the likely scenario for one of these 3 diseases to be introduced to the U.S. The second phase of this work is around domestic livestock, is around a domestic livestock model. So we use existing disease epidemiological model and information ascertained from the risk assessment to start a disease spread using the worst-case scenario. And domestic livestock shipments will be used, swine shipments exclusively for ASF and CSF, and then all 3 diseases, for FMD, all livestock movements will be included. And then for the last phase, we aim to add feral swine into this epidemiological model to see how the presence of feral swine impacts disease spread. A common comment that is made around this work is we know that these 3 diseases are very expensive, and so what difference does it make if it's 1 billion or 10 billion? We just need to prevent them. But that's not necessarily the case. Which disease has the potential to cause the most economic harm is the most important to prevent introduction. And so that could be moved into policy implications and management decisions around preventing illegal importation of specific products from specific countries. So that is a very important component. Another part that's important is that in the event of an outbreak, there's finite resources, and there's a number of differences between these 3 FADs. They are all costly, certainly, but there's an invertebrate vector species associated with African swine fever. There's vaccines available for CSF and FMD, but not for ASF. And FMD is a multi-species pathogen, whereas ASF and CSF are swine exclusive. And so it is important to have data and research around knowing how to manage limited resources in the event of an outbreak. And so with that, I'm wrapping up the targeted collaborations associated with the National Feral Swine Damage Management Program, and I'd like to move into continuing education opportunities that we have with our field biologists. The first was in December of 2018. We put together an African swine fever preparedness course. If you remember, African swine fever became hugely problematic on the global scene in the fall of 2018, and so we worked quickly to develop a course for our field biologists to bring them up to speed on African swine fever, clinical disease, and then the process for triaging samples and managing the potential of foreign animal disease on the landscape, and included a necropsy component. There's the happy group in Starkville, Mississippi of December 2018. We also hosted a course in Fort Collins in June of 2019 that was focused on foreign animal disease preparedness at large. The primary course objectives were related to the impact of foreign animal diseases. In specific, we focused on classical swine fever, African swine fever, and foot-and-mouth disease. But we also included other relevant production diseases, especially those that can present similarly to one of the 3 major FADs. We also talked fairly extensively about disease surveillance and diagnostics and hosted both a necropsy and field exercise components. The National Feral Swine Damage Management Program has a contract with Dr. Peter Fernandez, and he has been instrumental in our continuing education efforts. During the COVID times, of course, having in-person training is not feasible, and so he worked with Mississippi State University in partnership with the National Training Academy to put together a number of animal disease webinars that our folks can watch remotely to keep them abreast of the landscape related to foreign animal diseases. With that, I'd like to work into and begin to kind of wrap up with the interface with Veterinary Services. The National Feral Swine Damage Management Program, myself as the lead, works in very close partnership with Veterinary Services. I participate in the APHIS ASF Technical Working Group on a weekly basis, and then in the ASF Technical Working Group Planning Group on a weekly basis, and then have been intimately involved in ASF response planning in feral swine. Dr. Sarah Bevins, a researcher with National Wildlife Research Center and Wildlife Services side, was intimately involved in putting together the Red Book response about swine and kind of laying out the landscape for how Wildlife Services would approach managing ASF in the event of an outbreak. And then I also attend the monthly swine health meetings. We also have a number of collaborations that are going on and specifically related to work that has come up in partnership with Veterinary Services. One example of that is a hybrided Lyme project that's being done in close collaboration with Lori Miller and Rob Mickness related to carcass disposal and decontamination efforts. Specifically related to African swine fever virus, we know that getting carcasses off the landscape is really, really important. We've seen that in the European scenes. But we know from, managing feral swine is very different than domestic swine in terms of removal efforts and carcass disposition. And there are plenty of reasons to think that carcasses might not be moved immediately all of the time, even though it's a very important component of managing the disease on the landscape. And so we are currently in the throes of doing a pilot project related to hybrided Lyme. Hybrided Lyme is an extremely alkaline product. It's an agricultural product, but it can be used to create a sterile crust. And so the carcass underneath would still be, of course, heavily infected with African swine fever virus. But as long as that crust over the top remains undisturbed, the carcass's ability to transmit infection would be greatly reduced. And so in Missouri, Texas, and Louisiana, we've set up 3 pilot studies looking at the role the hybrided Lyme may play and the implication to scavenging animals to see if hybrided Lyme would serve to buy some time on the landscape to get those carcasses moved out. Another project that we have ongoing in partnership with Veterinary Services and came up as part of our working relationship is best practices for depopulation efforts. Early on in COVID, when a number of the swine slaughterhouses were shut down due to worker infections, and there became an issue with swine not having a place to go, there was talk about bringing Wildlife Services in to help depopulate those animals. Because our biologists get their hands on thousands and thousands of animals every year, we have used that opportunity to determine best practices for depopulation. So we're actually conducting research related to best practices. So what type of firearms, specific calibers, how far to stand from the target, whether, metrics related to humaneness of dispatch, and then, of course, whether the bullet travels through the animal or not. And this is really related to human safety and occupational safety in the event that Wildlife Services was called in to help detox animals. And Dr. Kurt McCottering's group is really taking the lead on this in partnership with Wildlife Services out of Texas. We have also partnered with Wildlife Services Maryland and their detector training dogs, working in close partnership with Marnie Pepper to make sure that the Wildlife Services detector dogs are capable of detecting feral swine carcasses on the landscape. This would just be another tool in Wildlife Services toolbox to find and remove carcasses in the event of a foreign animal disease outbreak. And then as part of the collaboration with Veterinary Services, it came to our attention that Wildlife Services does not always have an appropriate bank of ammunition on hand to respond to a foreign animal disease outbreak. And so we've been working with both Wildlife Services and APHIS leadership to develop an ammunition bank to be better prepared and to dispatch feral swine in the event of an outbreak. And with that, I'd like to begin to wrap up. I know I've hit you guys with a lot of information, and I'd like to step back for just a minute. When I started in my position 2.5 years ago, I never thought that I would have the opportunity to build and develop everything that has come forth in the last 3 years. I'm so proud of all of the work that we do, and really proud of the relationships that we have, and really feel like the Wildlife Services National Feral Swine Damage Management Program is a really integrated approach. We have a fantastic opportunity to partner with stakeholders both within APHIS and outside. We are a small headquarters entity. I run the disease component, and I have 3 fantastic support technicians that do the majority of national surveillance and help me on targeted projects. But because we're such a small, agile group, we're able to readily respond to the needs of stakeholder groups and really put a premium on relationship building. And so it's been a fantastic opportunity. I'm very proud of everything that's been built, and and really thankful for the opportunity. So with that, I'd be happy to take any questions. And again, thank you all for your time and attention, and Liz for the opportunity.
Thanks, Vienna. We do have a bunch of questions in the chat, so we'll go ahead and start with that. The first question is, are the surveillance blood samples banked?
So this, from every feral swine that's sampled, we pull 7 serum tubes. So we have 7 2 ml vials. So the first 3 of them are sent off to the diagnostic labs for the CRV, SC, and CSF testing, but we do retain 4 on hand as part of our archive. So for the majority of samples, we do have serum banked.
So the next one is, are there any plans to survey for ASF, a newly emerging disease?
Sorry, say that one again?
Are there any plans to survey for ASF, a newly emerging disease?
So we have the morbidity and mortality surveillance going on at the national scale, and that is, of course, just in relation to sick and dead animals on the landscape. And then the Puerto Rico project is looking at apparently healthy animals. There hasn't been any further conversation on testing apparently healthy animals on a broad scale as part of national surveillance. That we've had those conversations with partners in veterinary services, and I don't think we're quite ready to go down that path. So not at this time.
Okay. Are there any other collaborative projects in feral or farm swine populations in any of the other Caribbean islands?
Not that I know of, but there may be. I'm not sure. I guess International Services may have some information on that, but not that Wildlife Services is involved in, no.
Okay. Is there collaboration with private parties that offer services as hunting guides for feral swine?
Are there opportunities?
Is there collaboration with private parties that offer services as hunting guides for feral swine?
We haven't really done much of that. Michael Marlowe oversees the operational component, and so he may have some better insight on that. But no, we try and steer clear of feral swine hunting in general. The national program is pretty opposed to that. Because it often leads to encouraging anthropogenic movement of people moving feral swine from high density areas to create local opportunities in low density states. So we stay pretty clear of the hunting scene, but I'm not sure about the possibility of collaborating with hunting opportunities. So, Wildlife Services kills so many animals that getting our hands on pigs is not usually too big of a problem.
So we have a lot of thank yous for an informative presentation. The one question that's coming up a couple times is, are the Mississippi State University webinars available to the general public?
Unfortunately, they are not. So the National Training Academy is part of Wildlife Services, and all of the trainings are available for Wildlife Services personnel, but we haven't been able to figure out how to get those kind of isolated enough that we can have other personnel in there, unfortunately.
Yeah. And the next question is, how do we get more information on future collaboration opportunities or application processes?
So honestly, it's a pretty informal process. If you have a collaborative idea or an important stakeholder question, please reach out to me, and we'll determine how that aligns with the mission of the National Feral Swine Program. And but we're happy to partner, and we do it all the time. So just send me an email or give me a call.
Okay, this is a great thank you to you. Fantastic survey of all the activities Wildlife Services is leading on the feral swine front. The impact of these mammals on the landscape is significant in many ways. The next question is, why is there so many swine in North Dakota when there are none in adjacent states?
Yeah, so I think the map is a little bit misleading in that regard. It's just it was just a presence-absence map, but it is likely due to domestic swine breaking through their facility.
And the last question I have in the chat is if an ASF positive is found, what would be the response, knowing that a positive, whether true or false, would devastate trade from the swine industry?
So all of that is managed in really close partnership with Veterinary Services. I think that would all end up running through Dr. Shearer and Dr. Zach, and Wildlife Services would be providing support and personnel, wildlife epidemiological expertise, and some modeling expertise, and then of course operational removal. But all of that is really going to fall under Veterinary Services wheelhouse.
Okay. Do we have anyone else that wants to put a question in the chat? Oh, here we go. Why aren't these results shared directly with state-reported feral swine?
So on a quarterly basis, my staff took out an outreach map with the material associated with the feral positivity for both SB and TRV to our Wildlife Services state directors, and then they disseminate them as appropriate. And so I would recommend reaching out to the state director in your state, and I'd be happy to liaise that relationship or help out wherever I can. But those are provided on a quarterly basis.
Okay, and we do have a couple more questions coming in. Wildlife Services officials in each of the states know about or approve of the prevalence maps that you presented today. Wildlife Services cannot destroy them in some states without approval of other agencies. Have you seen the Canadian map of feral swine? It says a map of feral swine in the U.S. would indicate it's a miracle that only North Dakota has reported feral swine. That's a lot. I'm going to send this to you, and maybe you can write back to this person.
Okay.
And we will get all these questions from the chat. And someone asked me, where are you headquartered? You're headquartered in Fort Collins.
Yep. And my house now.
Yeah. Yeah, not at Wildlife Services, right? We're all just from our house. Okay, so with that, I'd like to thank Vienna, so much for doing this webinar. I thought it was great. A lot of information came through. So on behalf of the BS National Training and Exercise Program, thank you very much for presenting today. Our next webinar is Tuesday, February 2nd at 11 a.m. Eastern Standard Time. Lori Miller, who is a Senior Staff Officer with USDA APHIS, will share an overview of decontamination and disposal capabilities, gaps, and the status of projects currently underway, and priorities for the future. And just a note for everyone, if you have any ideas for webinars that the Vet Services and TEP can explore for our emergency preparedness community, please feel free to contact us. And you can find other webinars on so many other topics on the TEP video gallery online on the USDA APHIS website. And with that, I would like to say have a great afternoon, and thanks again, everyone.
Thank you.
That concludes our conference. Thank you for using Event Services. You may now disconnect.